A study that I recently reported on at the AACR meeting was a randomized phase 2 study of an adjuvant strategy that combined a neoantigen messenger RNA vaccine with a standard drug called pembrolizumab, which is an immunologic drug that's FDA approved as adjuvant therapy for high-risk surgically resected melanoma, compared to just the pembrolizumab alone. So this was a randomized phase 2 study with a 2-to-1 randomization that was designed to find out whether you could prolong the time to recurrence, or what we call recurrence-free survival, in patients who had what we call high-risk resected melanoma, meaning someone who after surgery would be free of disease, but would have easily a 50% or more risk of relapse over the next 5 years. This was a classic 2-to-1 randomization, meaning a total of 150 patients. So there ended up being 107 patients who got the combination of the messenger RNA vaccine 4157 with pembrolizumab, and 50 who got pembrolizumab alone. And remember, in a randomized phase 2 study, you don't have enough patients to definitively answer a clinical question, but you have enough patients to give you a high degree of assurance that if you did a randomized phase 3 study, a confirmatory study, the result would parallel that which you saw in the randomized phase 2 study. The results were highly encouraging and showed that there was a clinically significant difference in recurrence-free survival between the combination of the messenger RNA vaccine 4157 and pembrolizumab compared to pembrolizumab alone. Each arm had about 2 years of follow-up. And if you calculate, say, at 18 months, which is the so-called landmark recurrence-free survival, it was 78% for the combination and only 62% for pembrolizumab alone. Now, that 62% at 18 months is consistent with what you would see with pembrolizumab alone in prior studies, where it was compared in large studies to a placebo. 78% is pretty darn good. And if you look over time and compare the recurrence-free survival curves, you can generate a hazard ratio. And the hazard ratio was 0.56 over time, suggesting a 44% reduction in the risk of recurrence over time, where the recurrence-free survival curves were the same till about 8 months or so, and then began to open up and then stayed open over the 2 years of follow-up. In addition, when you looked at the toxicity, the so-called grade 3-4 immune-related adverse events, or the serious adverse events, were similar between the arms, although if you calculated all toxicity, obviously the vaccine carries with it its own toxicity. 89% of the patients who had vaccine toxicities, which is almost all patients, had grade 1 and 2 toxicities, which means they were fairly well tolerated. Only 1 in 9 or 11% of patients had a grade 3 vaccine-related toxicity, usually high fevers. But again, like the COVID vaccine that has some similarities to the neoantigen vaccine, you would have some vaccine-related side effects, as I've had when I got my COVID shot, and that is you'll see some feverishness, chills, fatigue, lethargy, injection site rash, and tenderness. And I mean, those are the classic muscle aches. Those are the classic vaccine-related side effects. So overall, when you add all the toxicities up, of course, there's more toxicity in the combination arm than in the control arm. But if you look at the clinically significant, the high grade, the serious adverse events, they were pretty much similar between the arms, and the vaccine did not augment the so-called immune-related side effects of pembrolizumab. I think the study was a landmark in that it's been the first reported study where a neoantigen vaccine approach, whether it be peptides, DNA, RNA, in this case, of course, messenger RNA, was compared to a control arm with a significant suggestion and evidence of clinical benefit for the vaccine when combined with the standard treatment of pembrolizumab compared to the control arm of pembrolizumab alone. All prior neoantigen vaccine strategies have been, or studies have been, early phase studies, very small numbers of patients, designed as proof of concept to see whether you could actually induce an immune response. Here, you actually had evidence of clinical benefit and significant evidence, clinically significant evidence of benefit. I think that makes it a landmark. And for the future, it says neoantigen vaccine approaches need to be pursued in other cancers that express neoantigens, which is almost all cancers, especially those where there's benefit from PD-1 blockade, such as non-small cell lung cancer, head and neck cancer, gastroesophageal, hepatocellular, renal cell. All of those are fair game for vaccine trials. Bladder cancer, all places where there's an approved adjuvant, or most of them have an approved adjuvant treatment consisting of, at least in part, a PD-1 antibody, where you could add the vaccine approach to it. And I think that will provoke large numbers of randomized phase 3 studies in the future. In addition, I think it gives hope to the neoantigen strategy in general, not just as a vaccine, but I think it now provides evidence in a randomized study that neoantigen approaches will be useful in cancer. And of course, it resuscitates the cancer vaccine field, in my opinion, and will provoke lots of further phase 3 studies. The cancer vaccine field has been discouraging. And I, for one, am certainly a person who would be regarded as a cancer vaccine skeptic. But now I think these data really shift the pendulum back to one where we really will wish to develop multiple different trials of neoantigen cancer vaccines, especially mRNA vaccines.